Good morning, good afternoon, everyone, and thank you for joining us today to Noxon Key Opinion Leader webcast regarding the top-line data of NOX-A12 in combination with radiotherapy in glioblastoma. This follows their presentation at ASCO 2022 last weekend. I am Guillaume Barentaghem from LifeSci Advisor, and I will be the moderator today. The webcast will feature a formal presentation, and that will be followed by a question and answer session. Should you want to ask questions, please feel free. You can do it at any time by just typing your question directly in the dedicated frame on the right of your screen. And once the formal presentation is complete, I will then read your question for Aram and Frank to answer the questions. As an information, this webcast is being recorded and a replay will be made available on Noxon Pharma's website as soon as possible. But without further ado, I leave the floor to Dr. Aram Mangasarian, CEO of Noxon Pharma, and as well to Dr. Frank Giordano, who is our key opinion leader today. He is chair and director of the Radiation Oncology Department of the University Hospital of Bonn in Germany, and is the lead investigator of the NOX-A12 GLORIA phase 1/2 clinical trial. Aram, the floor is yours.
Thank you, Guillaume. So thank you to all the attendees for joining us today. My name is Aram Mangasarian. I'm the CEO of Noxon Pharma. We're very happy to have Dr. Giordano with us today to take us through the research he presented over the weekend at ASCO in Chicago and give you some more details and his views on what this means. I'm just going to do a very brief introduction that summarizes some of the things that Dr. Giordano has laid out in his earlier webcasts about the therapeutic landscape in glioblastoma. You all know this is a very aggressive disease. 5-year survival very low, around 4%. Median overall survival for the full population is 12 to 15 months. Within that already pretty dire prognostic, there are some high unmet need groups, and these are the groups we're actually targeting in this trial. So there's a biomarker for the methylation status of a gene called MGMT, and when this is unmethylated, and you can test this before you start treatment, which is what we've done in our study, then you know that chemotherapy, the standard of care, will be ineffective. And if you look at the graph on the bottom right, you'll see the difference in the survival curves between the methylated in blue and unmethylated in orange population. And you'll appreciate there's a significant divergence between these two groups. The other group of patients that have a poor prognosis are those where surgeons are unable to fully remove the tumor mass. And so these patients also have because they have a larger starting mass of tumor, it grows back more quickly. And we're actually, in our trial, including both of these poor prognostic factors into the patient population. So that's just to give you a brief overview. And with that, I will turn it over to Dr. Giordano. Thanks, Frank, for taking the time to present to the people interested in our program. We really appreciate it.
Yeah, thanks, Aram. It's a great pleasure for me to present this data. I'll give you a bit of more background data on I'll go over the poster, walk you through the poster, but I also give you a bit of background information and a bit of an audio track that you usually don't see and hear once you are at the poster at these conferences. I'm very excited to lead this trial. We have been developing this idea based on long-standing preclinical evidence that was generated with A12 in autochthonous models or in autotopic models also. And based on that, I can just walk you through where we are with the clinical trial right now. So what I can basically now summarize, and what Aram touched upon, is already that we have started a clinical trial with radiotherapy and combined infusion of olaptaside. at 3 dose levels. Here, the recruitment is completed at the basically at this stage here, and that's basically what I've presented at ASCO, and that's what I'm going to present to you. What I can briefly touch upon is 2 cohorts that are now actively recruiting. It's expansion cohort with bevacizumab and 1 expansion cohort with pembrolizumab, and I'll probably will deep dive into that later and I'll give you then more information. Basically, both of these cohorts allow also completely resection. Okay, so we're talking about ASCO and a poster that we presented on ASCO. I'll walk you through it. We've numbered it, so you can basically download that poster also from the Noxon website. But it's a bit of a walkthrough here, and basically the background, very brief, is depicted here. Radiotherapy causes hypoxia. That in turn leads to 6-CL12 upregulation. That's the major molecule we want to target because that is basically mediating vasculogenesis and immunosuppressive tumor microenvironment through recruitment of these 6-CL4 positive cells. These cells are attracted by 6-CL12, and we're capturing 6-CL12 with olaptoside pegol, or I'm going to refer throughout the talk with A12. That substance A12, NOx-A12, binds and neutralizes 6-CL12. I already touched on the background and study design, so I'll briefly walk over that. It's a dose escalation part of the trial where we combined radiotherapy, which was chosen per investigator. By the way, we chose to include the true glioblastoma, the WHO grade 4, which you can see now on the Arabic letter here. The unmethylated Aram has already told you that this is a cohort where we have a high unmet need and where we know that we need a new therapy because chemotherapy doesn't work. We looked at safety as a primary endpoint. We've got a lot of secondary endpoints, which I'm going to walk you through all over the poster. What I'm really, really happy is that right at the start, we had a strong translational exploratory endpoint where we tried really to explore and to correlate the things we see also with histology and with biomarker imaging that were generated from advanced MRIs. So this is a bit of a background that I was talking about. I'm going to deviate a bit. I'm going to tell you that we have been looking on 10 advanced MRIs of these 10 patients, and we also had the chance to look on pre and post matched samples of 2 patients. But then we asked ourselves, what do we see there and what's different to what we see usually? And we thought it's a good idea to have a matched imaging control, which is matched per histology and also matched with this unfavorable biomarker, MGMT. And we needed to look into a codex reference, just that we know that what we see there, we can benchmark, and if we see that in a regularly treated patient population. Primary endpoint, I think it's the most important endpoint. This was basically a safety study, a phase 1/2 trial, basically looks at safety at first really primary endpoint. And what I can say here is that of all, if you look at that, we have 158 AEs, and most of them are not related to other therapy. Of those that are greater than or equal than 2, which were 77, only few were related to A12. You can look at those that are only A12 related. It's only 1 time a G3 at gamma GT and 2 times ALT increase, but only grade 3 and grade 2. So really a mild tox profile. If you compare that to the standard of care, that's absolutely a mild safety profile. When we started the trial, we said 26 weeks of therapy is sufficient and enough based on preclinical evidence. And then we treated these patients for 26 weeks. And what we then saw is that we should have been these patients on treatment longer. In some of these patients, that was just not enough. That we didn't know. And so we're looking at an early recurrence or a recurrence here once we stop treatment. In that patient, it turned out that this patient had a pseudo-progression here, so that basically looks like tumor, was resected, but was no tumor inside. And that patient also stayed stable in the cavity, and you see once we discontinued the drug a bit later, the patient recurred. In that patient, we also had the chance to look into what's going on here, where we didn't see tumor. We wanted to know what's in there, and we multiplexed that. I'm going to do that later. But the bottom line is that at that stage, we amended the protocol, and we then allowed to continue for a treatment as long as the investigators want, even beyond progression is possible. If you want to really figure out what progression is or not, in that case, we thought it's progression, but there was actually no tumor in it. So all our investigators are encouraged to give that drug as long as possible, as long as tolerated, which for the time being is no problem at all. And let's come to the main points where everybody's looking at is how do people respond, how do patients respond to A12? What you can see here on the left side is radiographic response. You may even see that we have 9 patients, but we have 10 patients in the trial. So we have both target lesions on the left side and non-target lesions on the right side. And what do we look at? So we take the sum of the target lesions, and we look at the diameters and we just add them up. And that's the left 2 images here. What you can see on the GLORIA patients, in A12 treated patients, 9 patients of all the 10 had a target lesion, which we could then follow along the treatment. And you see that 4 out of 9 actually had a partial response, something you rarely see in unmethylated glioblastoma. And 4 out of 9 had stable disease as a best response. But what does that mean, stable disease? Stable disease means you're not really touching the 50% tumor reduction. But as you can see here, these tumors actually responded, but they just didn't touch the defined 50%, which we would need to reach partial remission. But bottom line is almost all patients had one time point in which the tumor was shrinking, and 4 out of 9 where we had measurable target lesions even reached the threshold which is necessary to define partial response. Now, looking at the reference cohort, and I told you that once we look at the data, we were interested in how does that look in our other cohorts and how do they match. You're going to see that here. In that case, you don't see this image. You see it's a rather black and white image here. You see that few patients actually reach partial response. We looked at 20. Few reach stable disease, but the majority will just grow and grow further. And that we're talking about the best response under therapy, so that at no time point these patients were really having any sign of a response. We also looked at non-target lesions, and that's intriguing in that trial, because you definitely see a better response of non-target lesions to therapy than even to the target lesions. So what are non-target lesions? 9 patients had non-target lesions, which we could follow. These are usually located a bit further distant to the cavity. They are not really resected, and we can follow them, but they're not really getting surgery. They're not getting surgery resected, but they're also measured, and they're also in the radiation field, and they also get the substance. They're exposed to the substance. And what you can see here, that of all the 9 patients with non-target lesions, 3 had a complete response, 4 had a partial response. You see, they really almost touched the 50 here. Otherwise, it would have been a complete, also a partial response. And that one had a stable disease, but also decreased in size. To make clear how that looks like and why we look at it, I have an example. Oh yeah, here that's the exemplary case. You see, in the baseline scan, there's some residual disease in the cavity post-surgery. And here you see a non-target lesion, which is a bit more medially located here. So not an initial part of the cavity, not initial, what we looked at in the target lesion setting. We treated these patients with radiotherapy in A12, and you see how that really disappears over the course of treatment. And that is really intriguing, because if you look at the image, it's even a much deeper and better response of non-target lesions than the target lesions. We're really intrigued by that, and that's also a huge problem for glioblastoma, because these tumors tend to spread and invade. And at that stage, we actually have a surprising response of these non-target lesions. They even disappear. So when you look at the sum of non-target lesions, you might want to think of a dose dependency. If you look at the course here, we plotted this with the mean max change from baseline. You see that at highest dose it goes pretty well down. And also, if you look, you can dissect the tumor in several aspects, in highly perfused, medium perfused, and low perfused aspects. That comes out of the advanced imaging that we did here as a secondary endpoint. And what you can see, the fractional tumor burden, that's highly perfused. So these parts of the tumor that are really highly proliferating, highly perfused, they also go down with the dose. And then we have to see also in cellularity. We looked at the diffusion. That's also something you can measure. So the more diffusion, the lower the cellularity of a tumor. That means that when it goes up, it gets better. And there you also see a slight dose dependency. So we basically see the things that we wanted to see. We see a bit of a dose dependency in the target lesions, in the tumor fraction, also in the cellularity. Now I'm going to take you a bit outside of the poster, because we have also seen things that help us to really understand also what's going on. I told you that we have done surgery on a patient where we found a pseudo-progression, and now we're in the tingle-tangle between what is a pseudo-progression, what's a true progression. What we found out that under A12 we have a nice, if you want to wish, imaging biomarker that helps us to figure out what is true progression and what's pseudo-progression. Looking at that case, that patient here underwent resurgery, so we have the ground truth. What's that here? And if you look at the CBV, that means the perfusion. You see that every time you measure, even here, if you can't even see a T1 signal, perfusion already goes up a bit. And here you see the tumor coming back. Here didn't show his face, didn't really show hiding somewhere here, but already you see the biomarker going up. And also here, there's then the full-blown tumor recurrence, and that was then basically confirmed with surgery. So let's look at the case that I mentioned before, the case where we thought we have progressive disease. Look at what we see in the perfusion. No increased perfusion, even going further down along the path. And surgery confirmed that was no tumor. So with RCBV, we have an imaging biomarker where we can basically also understand the kinetics of the tumor. It's a very handy tool to understand the response to radiotherapy and A12. So last not least, I told you about Codex, and why did we do Codex? Because, well, in preclinical data, we know that A12 creates a really immunosuppressive tumor microenvironment, and that is basically repelling CD8 T cells from going into the tumor. This tumor microenvironment is really repelling these cells. And what we see under A12, what you can see here, because we looked at the CD8, this is the T cell, the cytotoxic T cells. Granzyme B positive means they are active. Ki67, that means they are dividing. So this is the fraction, if you want to wish, of the aggressive anti-tumor T cells. You look at the baseline and you look at A12, you really see that this nicely goes up. If you see the images here, see that here you don't really see that in that manner, but you see these clusters here. Also here you see T cell clusterings. That means here there's a proliferative nest of T cells going up. And also with the macrophages, we've seen things we have not seen before. For example, I wouldn't say that there's pretty much a change in macrophages, like from the numbers, but what we've seen from the configuration of the macrophages, this is a patient that did not respond to therapy, and there we see some cuffing of macrophages. They really encapsulate T cells and keep them hostile somewhere here, things we have not seen before and that are not described essentially, and we're now deeper looking into that. So just to conclude that, we think radiotherapy plus A12 in a population of chemotherapy resistant or refractory population is safe. We did not see any DLT. I think we see a lot of clinical efficacy signals, more than expected, with target lesions and non-target lesions even more. We see T-cell recruitment and clustering. We even see what happens if macrophages encapsulate these T-cells, things we have not seen before. And now we even go deeper, and I've also mentioned in the previous slides that we're now combining it with bevacizumab and pembrolizumab, so we're targeting VEGF also and PD-1 axis as a consequence of what we've also seen in this set. With this, I hope I could make clear like what we've presented at ASCO, and also figure out what we see in the imaging biomarkers. And I'm happy to take your questions, Guillaume, if there are any, or if any of your participants has a question, you can also put that in the chat. I think we can probably then answer it.
Yeah, thank you very much actually for your time. Just as a reminder, if you want to ask any question, you can do so actually with a box called Q&A on the right of your screen. We already have a few actually. I mean, we have a few actually which relate to questions unrelated to NOXA12. So since it's a KOL event on NOXA12, I hope you don't mind if we solely focus on the NOXA12 questions. For the question outside NOXA12, if you could just send them to me or to Noxon Investor Relations team, and we'll try to address them offline. One of the questions actually, which was in the package with question unrelated to NOXA12, was regarding the next step with NOXA12. What the next step are going to be with regards to the clinical development?
You're on mute, Aram.
Thanks. Yeah, so I think as Frank outlined, we're going to complete these extension arms, and I think the data emerging from those is going to be quite important in determining the next steps and what we actually take forward. And so our goal now is to give an update on that and the strategy for developing it in the next few weeks. So I think I'm going to have to push the final response to that question off a little bit.
Okay, thanks. We have another one with regards to why not extend the treatment beyond 6 months with a low dosage of NOXA12? Why not combine NOXA12 and NOX-E36 in patients resistant to basic treatment? And actually another question again the same vein, when are the interim and final results expected for the expansion of the GLORIA study? I guess referring to the combo with Avastin and with PD-1.
Yeah. Well, a lot of good questions. I think you saw both of us smiling because we're thinking a lot about those very combinations. I think for the moment we're focused on getting the data out of the bevacizumab, so the anti-VEGF arm and the anti-PD-1 arm. The anti-bevacizumab arm is moving more quickly, as it's got a slightly easier to fulfill inclusion criteria than the other arm. And I hope to be able to give a high-level update on where we are again in a few weeks, because that's going to influence the path forward. Frank, do you want to comment on these other ideas for combinations and so on?
I think they're brilliant ideas, and I think they're brilliant ideas. Maybe I can add that we started the trial like with the idea of like what came out of the preclinical models is that we have a like anti-vasculogenic effect, and I think I can say that we're stunned about the things we see on the codex, on the histology level, because that opened many other avenues, and also what we see in the advanced imaging. We really had, and thanks to Noxon, you went that way with us. We really put a lot of efforts in the imaging just to understand the various aspects of these tumors. And I think I'm not telling too much that we have good ideas coming out of that now, of that very, very helpful phase 1/2 trial.
Yeah.
Okay, thanks. We have as well. Yeah, sorry.
It's fine.
Yeah. We have a few questions as well on the RCBV. mean, this slide actually you mentioned with regards to pseudoprogression. I mean, so one of the question is, let's do them one by one, but what MRI sequences did you use for RCBV?
Dynamic susceptibility weighted imaging. It is a T1 star weighted sequence. You inject the contrast agent and then you do frequent scanning with T1 star sequences, and then you just look at the susceptibility curve and you see if you normalize that to the healthy brain on the other side, you can see that you basically have a great relative perfusion number. RCBV is the number, is the term that we're looking at. Okay. And then you dissect the tumor in RCBV values, and then you can dissect the tumor in high, medium, and low perfused aspects.
Okay, and so in the same vein, so the thing is that, okay, the RCBV analysis is interesting. Do you plan on using RCBV as a kind of surrogate endpoint in your pivotal trial to allow the trial to continue in case of pseudoprogression? And as well, was this phenomenon of RCBV repeated in all the pseudoprogression that you observed?
Guillaume, look at that. I don't know how many active substances in this world were on the market that were not successful because they were too early discontinued. Look, if you have, imagine you have an active substance and you have a lot of pseudoprogression, you're going to discontinue active therapy because of a false friend information, and the patients are going to die. And at the end, you're going to say, oh, well, yeah, that was a progressive disease anyway because the patient then subsequently died, but the patient died because you de-seed the therapy too early. I don't really want to imagine how many patients have really not been benefiting from active therapies because we didn't have really good tools. We're now in a situation where we have a quite good biomarker to understand where we have a chance or where we see early progression. We also have a patient that I've shown you that hasn't really responded to therapy. So we're not like 100% winning hands down, but we now have a biomarker that we can maybe use. If it's the pivotal biomarker, I don't think so. For a pivotal trial, you need hard endpoints, but you got to make sure that to get to that hard endpoint, you don't DC an active therapy too early. And there, RCBV is pretty helpful.
Yeah.
Okay. We
want to make sure that, you know, in the pivotal trial, and this is one of the things that we've been discussing with Dr. Giordano, is that the centers know to look at this perfusion value before they discontinue therapy, because that's going to be key to make sure we reveal the true efficacy of the substance.
Okay, and just have a silly question from me, but I can assume that any medical center in the pivotal trial would be able to use such a tool, correct?
I mean, every center that's participating here is a certified brain tumor. Like they run through various certifications, and they have neuro radiologists or radiologists that are qualified to look at that. And to be fair, we didn't invent the wheel. Perfusion imaging is I could say, in 90% of brain tumor treating centers, a standard sequence that's run to distinguish that, to help distinguishing pseudoprogression from true progression, because you can also use it as a response classifier in, for example, in brain mets that are where patients are under immunotherapy. You have the same situation. These brain mets initially tend to get larger after radiotherapy, and then they shrink. And their DSC is pretty much established.
Okay. Next question: what were the criteria used to the matched cohort?
So we tried for the imaging to look at matched cohort in terms of, and we cannot compare MGMT methylated that all respond to temozolomide with an MGMT non-responding population. So the key point where we matched it was that it's unmethylated MGMT, these tumors that do not respond to any of the therapy we offer the patient. So that was the key matching parameter. I must also say we also had a positive selected the imaging cohort had to have at least 2 or more scans in the follow-up. That means of all the patients we looked through, all those that died early after maybe surgery, radiotherapy, and that didn't even reach the first scan, we didn't look at that because we needed some scans at least to figure out what the best response are based on 2-3 scans. So the matched control cohort, if you wish, is a bit of a positively selected group of not so well performing patients.
Fair enough.
So
it was clear. But pretty difficult to explain.
Maybe I can just restate that. So you're actually excluding the worst performing patients.
Exactly.
So you've got the patients who survived long enough to have multiple follow-up scans.
Exactly.
So it's a slightly better performing cohort than you would expect from simply applying the recruitment criteria of our trial.
Okay, very clear. Next question. Have you seen any interesting quality of life improvement in treated patients?
We're measuring QOL with the standard questionnaires, but so far, what I can say is that if a patient is diagnosed with GBM, you don't really try to improve QOL. You try to maintain QOL at the level of baseline, and there we didn't see a decrease under A12. You definitely see a decrease in QOL in the moment that there's tumor progression or there's anything coming along the path that is tumor related. But considering the therapy, I can say even like with the needle and like with replacing that the substance needs to be 24/7 infused via pump, I have not seen any or one patient that would have had a decrease in QOL because of the circumstance of the application of the drug. People are definitely willing to do that because it may be a bit unhandy to carry a bag with the pump, but people are willing to do that. And I've not seen a decrease in QOL so far under therapy.
Okay, next question. Does Noxon intend to explore to participate to a platform trial for the phase 2/3 like the GBM Agile study?
Yeah, that's one of the platform trials that we are indeed looking at. That's one of the options. We haven't made a final decision yet. I think we need to see how the data shapes up. For those of you who don't know what GBM Agile is, it's a cooperative platform trial where there's a shared control arm, and then multiple active agents are tested experimentally. And it's nice in a few ways. It comes with some limitations, which I'll get into in a minute, but it's nice because it's up and running. It allows you to join a trial that's running in the US, Canada, Europe, and soon Australia and China that would lead to approval. And so you can be up and running quickly. The limitation is that you have to adhere to the standard protocol. You have to adhere to the standard imaging. You can add some, and that's one of the things we're exploring with them, is whether we can add these important parameters that we've understood through this very important early work that we're doing. Is important to keep treating patients when they are actually responding, but the imaging looks like it might be a progression.
Okay, very clear. Probably a question for you too, actually. Why did you choose to go for BEV and PEM?
Bevacizumab and pembrolizumab?
Yep.
I think that's Frank. I think you wanted to.
Okay.
You can take that.
So I get like when you look at all the preclinical data we had, the BEV combo is a logical next step because hypoxia leads to HIF1 alpha, hypoxia-induced factor 1 alpha, mediated upregulation of CXCL12 and VEGF. So that is well known and established for a long time. And one is the salvage pathway of the other. That means if you inhibit one, you may want to circumvent that effect with VEGF. And it was shown that the VEGF blockade could be circumvented with 6CL12. So inhibition of both has already been assessed in a murine model, and that was seen to be synergistic. But we had to start somewhere and have, like, first of all, wanted to see if there's a dose dependency, if we can establish that, if we can make that work, and is that toxic at all, combine it with radiotherapy. But the BEV combo was definitely there, like a logical step from all that's known in preclinical models.
Yeah.
The motivation to include pembrolizumab, I hope I'm not going abroad with what I'm saying, is, but that was not on our watch, like when we started the trial. But when we looked at it with Codex, and then with Codex you can look at 50 biomarkers at a time at the same slice that you look at. We saw T cell clusters, we saw massive influx of CD8 positive, so the effector T cells, and that was great. And that was not only seen in the 2 patients that I mentioned, but it was also seen in another trial of Noxon. It's called the OPERA trial, where A12 was given in metastatic colorectal and pancreatic cancer. So there was now accumulating evidence from the studies that are ongoing. It definitely makes sense to combine it with immunotherapy. But to be fair and honest, I think we all didn't have that on our list, and it's something where I'm really happy that we had a strong translational component up and running in that trial.
Yeah, I'll just add that the images, if you look in the publication of the OPERA study, are really very similar to these clusters that Frank and his colleagues found in the brain cancer tumor tissue. You see these T cells going from sort of dispersed all throughout the tumor to these clusters that are very close to the cancer cells. And actually in the pancreas and colorectal cancer tissue, that was one of the statistically significant outcomes, was that the T cells actually moved closer to the cancer cells in the tissue in the patients who had this tissue response. So it's a very interesting phenomenon. Looks like it's A12 driven, because in that earlier trial in pancreas and colorectal cancer, there was no radiation. And this before and after samples were only after 2 weeks of NOX-A12 monotherapy.
We have a next question from Jamila at Invest Securities. I mentioned her since she covers your stock. How should we consider NOX-A12 as an agent that optimizes radiotherapy anti-cancer effect, or rather like an inhibitor of the latter effect of radiotherapy known to be pro-cancer?
Oh, good question, Jamila.
I don't know if I got it right, but like radiotherapy has been used in glioblastoma for decades, and we've tried to dose escalate, we've tried to concentrate on the cavity, we've tried everything, hypofractionation, hyperfractionated, accelerated, we tried everything. What we've learned so far is that we can be quite effective with radiotherapy, but we need to make sure that after we're done with radiotherapy, these tumor and these microenvironment will recover quite quickly. And what we've learned over the past 5 to 7 years is that radiotherapy might be beneficial in depleting tumor cells, but it also depletes small vessels. And the response of the tumor microenvironment is something you don't want to have. And over the past years, we've understood that we have to block certain pathways, or we have to modulate the post-radiogenic tumor microenvironment. So A12 is, to my understanding, not a radiosensitizer, but rather like an effect stabilizer, if you wish. Like you deplete the vessels and you kind of conserve or preserve the status quo that you have here after radiotherapy.
Yeah, I think that this blocking of repair of the destroyed vessels as one of the key effects, and the earliest preclinical work done at Stanford on this effect really focused on the vessel depletion. And I think, you know, why Frank mentioned earlier, we weren't focused so much on the immune response, which is the other aspect we're now seeing in these tumors that's quite interesting, is that that wasn't analyzed as part of these early studies, which were very much focused on the vasculature. This came out of work we did in pancreas cancer, and we weren't sure, to be honest, that we were going to get this influx of immune cells into the brain because it's an immune privileged site. But we saw it, and indeed we saw it at the lowest dose already, in pretty dramatic fashion. So it looks like we have two effects. One is this blockage of repair of the damaged blood vessels, and the other is this enhanced influx of the active arms of the immune system.
Next question. For now, the GLORIA treatment paradigm is radiation followed by continuous NOX-A12. Should the patient progress after, let's say, 6 months, how would you expect this patient to be treated? Would that be by additional radiation and starting NOX-A12 again? I mean, basically, how do you see treatment post-progression, if any?
So first of all, we have to make sure that it's a true progression. I think we've touched on that pretty extensively. But if we find out it's a true progression, we are, so to speak, mechanistically, all the investigators are tempted to give bevacizumab because you feel that maybe that was the backup pathway that was activated. And we all know, I've showed a patient, we all know that salvage temozolomide in the unmethylated patient will essentially lead to nothing. These patients will not respond to temozolomide, so you gotta have a better idea. And based on, I would say, preclinical evidence and based on everything we know from these models, we are tempted to give bevacizumab. But to make that clear, I could also imagine recurrent radiotherapy and continuing A12, because you can back deplete the vessels and sort of delay that again. But that's all speculative, and in the recurrent setting, we don't really know what's going on, and if one or the other way may be beneficial, I can't really. But I could think of a re-irradiation, definitely, and trying to tackle that again, maybe like with a combination or
And so would you even expect actually a total new cycle with resection, radiation, NOX-A12, plus or minus Avastin then? Beva.
Well, all highly speculative. I mean, we're not so far that I could really give a quite fundamental, scientifically based answer to that. I mean, if a patient is able to be re-resected, I would always prefer re-resection because you get the ground truth. And I would even think of, like, based on the experience we made, I would even think of a low threshold for stereotactic biopsy to really nail that down if you are dealing with a true progression or with only pseudo progression, because these are two totally different ways of treating and two totally different consequences for the patients.
Okay. In your slide, we can see that one patient has GGT increased and two have ALT increase, only NOX-A12 related. Can you say when this increase occurred, open bracket, early or late in the treatment, and if there were any correlation between this liver elevation and poorer outcome in these patients?
No. So wait, both patients we have included had pre-existing diseases. Both patients with liver enzyme elevations had pre-existing diseases that may per se cause these liver enzyme elevations. For example, one had chronic diverticulitis and the other had idiopathically elevated baseline liver enzyme level. So we understand from earlier phase 1 trials on H12 that there were liver signals coming up, but these trials had much higher exposure doses. Aram, correct me, but we're definitely paying attention. But these previous trials with the liver enzyme signals had much higher doses.
Yeah, so I think it's one of the reasons that the investigators are so sensitive to seeing liver enzyme elevations, and by precaution want to flag it as probably NOX-A12 related, even if there is a pre-existing condition. It's something we're keeping a careful eye on as a company. I know the investigators and treating clinicians are keeping a very careful eye on for the patients.
Thank you. So the next question, how challenging the recruitment of these patients was, and if the data are confirmed in phase 3, how likely it is for NOX-A12 to become standard of care in this patient population? And maybe actually, can you remind actually how big this patient population is?
So GBM has an incidence of 3 to 4 in 100,000. 60% are unmethylated. You do the math. And I would say it doesn't matter if you have a resectable or unresectable tumor. I mean, it's both the same. In the first few patients we included unresectable. In the first dose levels we included unresectable, and then we now opened the trial for fully resected. I think recruitment is always an issue with glioblastoma, because only half of the patients are operable, and with those that are then unmethylated, you only get a subfraction of a very rare disease. I think the point is that in high volume tumor centers like we have in Bonn, or all the sites that are participating, that is not really the issue. The issue is to find a patient that is not so bothered by the initial status that you really have the chance to help these patients with a therapy. I'm making an example of patients that is under permanent seizure with a tumor in a very delicate location. You know, these tumors, they even may develop pseudoprogression and die then from a pseudoprogression, or have any complication that you cannot control. In all the clinical trials with glioblastoma, patient selection is absolutely key to find, or to even have the chance that you can really figure out an effect from like an unspecific background noise that this disease really creates. And the second part of the question, I mean, that's the sort of jackpot question. If you have a positive phase 3 trial, you're definitely your water falls on dry ground. I mean, there's nothing been changing this therapy for 20 years. So if you come up with a positive signal in a phase 3 trial, then you can basically check the guidelines all over the world. That's going to be standard of care immediately.
Okay, so question to Frank specifically, that says the proportion of drugs developed in GBM with positive phase 2, but failing to demonstrate clinical benefit in phase 3 is very low. I think we can agree.
Yeah.
How would you position the robustness of the NOX-A12 clinical data generated to date with the other phase 2 generated by other compounds, and what do you think makes NOX-A12 so different?
Yeah. Well, it's a very good question. And actually, if you go to a pharmaceutical company and try to convince them from like going into glioblastoma, you will have to face this question. And you definitely have a prepared Frank, because this question is the key question if you want to get in touch with a pharmaceutical company. But the key is that you have to implement an elaborated and sophisticated imaging protocol. And in all earlier trials that I know, this was not really implemented. I already mentioned, I don't know how many active substances are really burning in the fire of pseudoprogression, and where the therapies get discontinued despite having an efficacy signal, which you don't see because you don't measure the right thing. So in other words, we now and today know better if we deal with pseudo-response or pseudo-progression and can figure out how we deal with one or the other. And one very, very important point in that trial is that we looked into the samples with massive parallel immunoplexing, that is looking at 50 markers at a time, just to make sure that the things we see with the imaging correlates to a kind of histology, and that is actually the case in that trial. So the plan was not to go in a naked phase 1 trial and see what happens. The plan was to go in a phase 1 trial, establish as many as possible translational components to get the wider picture that is definitely necessary to decide whether or not we move on in the clinical development or not. And that's where we right now are.
It's a really important point, and I think that the work that the group that Frank has assembled around the trial, not just the clinicians in the clinic, but also the scientists looking at the tissue, has been really extremely important in advancing our understanding of how NOX-A12 works.
Okay. I think another question for Frank that requires is crystal ball. Based on your experience, what kind of median OS we could expect based on the ORR that you are achieving in the GLORIA study?
I'm deeply looking my crystal ball. Like, I cannot really comment on that because follow-up is not mature yet. There's 2 out of 10 patients that are still on drug or in follow-up. But what I can say, I don't know if I can say that, but 50% of patients achieve to live already now longer than a year. And there's 1 patient on trial that is now close to a year. So we're going to be exceeding 50%. But these efficacy parameters will take a bit more time. Also, I have to mention, be careful if you look at classical efficacy parameters, because I showed you an image of a patient that we discontinued, or 2 patients that we discontinued too early. And that was a steep learning curve we all took in the GLORIA trial. And these patients probably could have lived much longer if we would have kept them on the drug for longer than 26 weeks. So we're looking at the learning curve. We're looking at a not mature follow-up, and all these things come then together. And believe me, I'm having a hard time in putting that together, putting that down in words in a manuscript that describes all that.
Yeah. But maybe a follow-on from me. Can you remind us, in the unmethylated population, what is the median, so not the median OS, but the 1-year survival rate? Because you mentioned we may pass the 50% for the GLORIA.
Yeah, I think the median is, Aram, go ahead.
Yeah. Well, I think it's not just unmethylated, right? Because we're taking unmethylated and incompletely resected, right? And when you put those 2 together, I mean, you're getting the worst half, and then you're taking the worst of that. I think the best publication we found, and the one we always cite, is the study that broke it down the best, and that came to around 10 months OS for that population. And Frank, I think from your clinical experience, you should say whatever you think.
Yeah. A benchmark from the trial where you showed overall survival data, Aram, this was the ERTC trial where MGMT was also established as a biomarker, and where temozolomide was established as a therapeutical drug, but where you have to be sure that those, per today's definition, were not really all true glioblastoma. At that time, there was also a huge fraction of IDH mutated tumors that would not be qualifying as glioblastoma, and there the 12-month survival was about 40%. They had 80% resection rate. So we're looking at, like out of the blue, I would say we're looking at 25 to 35 something percent, 40% 1-year survival if we would match the whole population. But since our trial is not randomized and we can only like make up some numbers, but like I would say we're not so bad, even though we had a learning curve and we had discontinued many patients too early.
Next question: How do you explain the A12 effect on non-target lesions? Open bracket, recruitment of immune or inhibition of vasculogenesis?
Okay, I'm going to take my glass bowl again here. I think the point is that where we have a low tumor volume, exactly that, I think the question goes right in the right direction. And we discussed that many times, where you have these low tumor volumes where actually the immune system can win. If you look at all the immunotherapy trials, they want to have a low tumor burden to start, so that basically you can win. We also have a good idea why these non-target lesions really can be disappearing, but at that time I have no proof that it may be immune related, or we don't really have the proof for that. But it's really making us wonder why these small, so to speak, abscopal lesions would then disappear. It may well be that they're small enough to be defeated, so to speak.
Okay, there's a kind of quality of life question here, which says NOX-A12 is a permanent infusion. So did any patient discontinue who had convenience-related issues? I mean, leading to some not optimal treatments. Is there a way to formulate NOX-A12 as a more convenient formulation that will not require a pump, such as a weekly subcutaneous injection?
I'll let Frank answer the question about patients discontinuing, and I'll take the one about
or not fully optimizedly treated in an optimal manner. I mean, were we sure that the dose of NOX-A12 was absolutely flat and stable?
Yeah.
So when it comes to convenience, I would say these patients get a deadly diagnosis, and whenever these patients, they basically try to do everything to come into the trial, because the alternative is definitely to have no therapy option at all. So for the time being, I have not experienced these patients to be protesting against the pump. Rather, to the contrary, the patients where we discontinued after 26 weeks before we amended the protocol I think they were pretty close to coming to Berlin and to protest that we take away the pump, but we were simply not allowed to make that infusion longer. When it comes to convenience, I see that this can be solved in the future. I mean, for GCP reasons, a trained staff has to replace the needle and the pump. This is just for study and GCP reasons. I don't see why this could be a problem, that the patient just disconnects it and connects it. I mean, like many cancer patients do that themselves and replace the cassette themselves, so they won't even have to come in the hospital to do that. You give them the pump on stock. I don't know if there's an issue with how stable the substance is, but to my notion, this is very stable in room temperature or even warmer environments. I see no problem why the patient cannot change everything once weekly. But with the formulation, I don't know, Aram, that's up to you to comment on that.
Yeah. Maybe I'll make a few comments. I mean, you're correct on the stability. Indeed, we don't have any issues with stability of room temperature, and we've even thought about loading the pump with enough drug for longer times between refilling. The reason we use the pump is really to have a completely flat PK curve, because that's what was seen in the early models in the rats when you give a subcutaneous administration. We may explore something, we're thinking about other dosing regimens that involve IV bolus administration. That way the patient won't have to carry anything around between. But at the moment, that would have to be more frequent. We're also looking at formulations where you would have a delayed release, and so you would essentially the goal of that research would be to get a formulation where you have a subcutaneous or intramuscular injection, and then the drug is released, and you get a similar curve to the pump. But again, that's something that would have to come later. We're looking at various formulation options that are available, and that's something we would definitely think about.
Okay, but I think we are up to the time. So Frank, Aram, thank you for your time. Again, if you have any question on the call, please feel free to reach out to us. If you have any additional question to Frank as well, please feel free to send them to us by email, and we'll be happy to answer them as well as possible. So thanks again. Have a lovely weekend. If Aram, you have any word for the end?
No, thanks, Frank, for heading the trial and participating in today's call. Guillaume, thanks for the organization. All of the attendees, thanks for your attention. Look forward to interacting more with you in the coming weeks on the future plans.
Thank you very much. Have a great weekend all. Bye-bye.
Bye all.
Bye-bye.